Virtually all the reactions in the body are mediated by enzymes. Especially metabolic processes fully depend on enzymes. Therefore, understanding the basic property of enzymes is prerequisite for understanding metabolism. Today, we will cover 7 topics. But I would like to highlight topics 5 and 6. Michaelis-Menten Kinetics and enzyme inhibition. These are the high-yield topics for USMLE. Enzyme can be divided into 6 classes. First class, oxidoreductase, catalyze oxidation reduction reactions such as oxidation of lactate and reduction of NAD+ by a dehydrogenase. Second class, transferases, catalyze transfer of functional group such as a methyl group from the serine to folic acid by a transferase. Third class, hydrolases, catalyze cleavage of bonds by addition of water, such as cleavage of urea by urease. Fourth class, liases, catalyze cleavage of bonds directly, such as cleavage of carboxyl group by a decarboxylase. Fifth class, isomerase, catalyze structural rearrangement of groups, such as rearrangement of carboxyl group by a mutase. Sixth class, ligases, catalyze formation of bonds at the expense of energy, such as addition of carboxyl group by a carboxylase. Each enzyme has its unique identity number or EC number. The first number represents the class of the enzyme. We only need to pay attention to the first number. When you are given an EC number, you are expected to know which class this particular enzyme belongs to. To help you remember the name of the 6 classes, here we use a mnemonic. Old Testament heroes lied in life. O represent oxidoreductase, T transferase, H hydrolase, L liase, I isomerase, L ligase. Now let's go to part 2. Any enzyme should have an active site in which a particular reaction takes place. The active sites can be further divided into binding sites and catalytic site. The enzyme uses binding site to grab the substrate and use the catalytic sites to make or break the substrate. Specificity refers to the fact that an enzyme only recognizes a specific chemical compound. A cofactor refers to an inorganic component for an enzyme, such as metals. Coenzymes refers to organic but non-protein component for enzyme, such as vitamin C and vitamin B6 you have seen for collagen synthesis. Holoenzyme refers to enzyme plus cofactor and or coenzyme. Apoenzyme refers to the protein part of the enzyme without cofactor or coenzyme. Proteolytic group refers to a non-protein component that is very tightly attached to the protein. It can be inorganic or organic, such as vitamins and heme. Most enzymes are localized in specific locations within the cell. For example, DNA polymerase resides in the nucleus. Enzyme in glycolysis resides in cytoplasm. Now, let's look at part 3. How do enzymes work? This question can be broken down to 3 related questions. What does the enzyme do to the reaction? What does the enzyme do to the substrate? And what does the substrate do to the enzyme? As we know from organic chemistry, any chemical reactions undergoes a transition state before the final product is made. From reactant to the transition state, there's an energy barrier called free energy of activation. Enzyme lower the free energy of activation. What does the enzyme do to the substrate? Enzyme first combine with the substrate to form an enzyme-substrate complex. The complex goes through a transition state. Enzyme stabilize the transition state. As a result, the enzyme increases concentration of the reactive intermediate that can be converted to product. Substrate is converted to product, and then the enzyme product complex form. Finally, the product is released. What does the substrate do to the enzyme? There are two models to explain this. One is an early model proposed by Fischer, lock and key model. Only a substrate of the proper shape could fit with the enzyme. In this model, the substrate doesn't do much to change the enzyme. This model could explain only some aspects of the mechanism of the enzyme, but it is simple. We will use this model to explain competitive inhibitors. Second model, induced fit model. Substrate can induce a change on the enzyme. This model can explain most aspects of the mechanism of enzyme. We will use this model to explain non-competitive inhibitors and uncompetitive inhibitors later. Now, let's move to part 4, factors affecting reaction velocity. Velocity refers to change in concentration of substrate or product per second. Factors affecting velocity include substrate concentration, environmental factors such as temperature and pH, and the presence of coenzyme or cofactors. Velocity increases with substrate concentration. Here is a picture shows you two types of enzymes. Enzymes follow Michaelis-Menten Kinetics in blue shows hyperbolic curve. Allosteric enzyme shows sigmoidal curve. Velocity increase with substrate concentration until a maximum velocity is achieved. Each enzyme has a specific optimal pH. For example, pepsin has an optimum pH around 2, and trypsin has an optimal pH around 7. Enzymes typically have an optimum temperature around 37 degrees Celsius. Now, let's go to part 5, Michaelis-Menten Kinetics. This is the important part of this lecture. Kinetics is the study on how reaction velocity change with substrate concentration. For Michaelis-Menten kinetics, we will use initial velocity or V0. V0 is a change in reactant concentration as soon as the enzyme and substrates are mixed. Michaelis-Menten kinetics is based on the model. Here, E represents enzyme, S for substrate, P for product. Enzyme reversibly combine with the substrate to form an enzyme-substrate complex. This complex irreversibly breaks down to product. This model is based on 3 assumptions. First, concentration of the substrate is much greater than the concentration of the enzyme. As a result, only a small fraction of the substrate is bound by the enzyme at any given time. Second, the concentration of enzyme-substrate complex remains steady over time. Third, only V0 is used in the analysis. Michaelis-Menten Kinetics describe the relationship between initial velocity and the substrate concentration. It states initial velocity V0 equals to Vmax times concentration of substrate divided by Km plus substrate concentration. Here, Km and Vmax are constants for a given reaction. Here we ask, how can we determine the two constants, Vmax and Km, when the kinetics of a reaction is given? Vmax corresponds to the maximum velocity by the system. Km is equal to the concentration of the substrate required to achieve half Vmax. Michaelis-Menten kinetics has several features. Enzyme with a small Km has a high affinity for the substrate. Here are two enzymes for the same substrate. The one with a small Km in blue has a higher affinity for the substrate than the other enzyme. When substrate concentration is much lower than Km, velocity of the reaction is approximately proportional to the substrate concentration. On the other hand, when the substrate concentration is much greater than Km, velocity is almost constant. Third feature, velocity is directly in proportion to the enzyme concentration at all substrate concentration levels. Lineweaver and Burk manipulated the Michaelis-Menten equation by taking its reciprocal values, generating a double reciprocal plot. Here, x-axis represents reciprocal of substrate concentration, and the y-axis represents reciprocal of V0. Michaelis-Menten's equation now become a linear graph. X-intercept corresponds to inverse of Km with a minus sign, and the y-intercept corresponds to inverse of Vmax. Now, let's move to part 6. Many substances can inhibit enzyme activity, such as substrate analogs, toxins, drugs, metals. Inhibition can be divided into two broad classes, reversible inhibition and irreversible inhibition. Reversible inhibitors form weak non-covalent bonds with the enzyme. Examples include many drugs. Irreversible inhibitors form covalent or very strong non-covalent bonds with the enzyme, usually permanently inactivating the enzyme. Examples include heavy metals, aspirins, and organophosphates. Enzyme inhibition can also be classified by the way the drug competes with the normal substrate. Competitive inhibitors resemble the normal substrate and compete for the same sites. In competitive inhibition, the Vmax remains unchanged, but the apparent affinity for the substrate is reduced. Examples of competitive inhibitors include methanol that compete with ethanol for alcohol dehydrogenase. Methotrexate competes with folic acid for dihydrofolic acid reductase. Captopril competes with angiotensin I for angiotensin converting enzyme. Non-competitive inhibitors bind at a location other than the active site, which results in a change in the enzyme activity. In non-competitive inhibition, Vmax is reduced, but apparent affinity for the substrate remains unchanged. Examples of non-competitive inhibitors include physostigmine and allopurinol. Uncompetitive inhibitors bind the enzyme substrate complex or ES complex at the sites other than the active sites. It increases the apparent affinity for the substrate but decreases the maximum enzyme activity. For example, tertiary amines are uncompetitive inhibitors of acetylcholine esterase. In summary, in competitive inhibition, Km is increased, but Vmax is unchanged. In non-competitive inhibition, Km is unchanged, but Vmax is decreased. In uncompetitive inhibition, both Km and Vmax are decreased. In Lineweaver-Burk plot, this line represents Michaelis-Menten kinetics without inhibitor. After adding a competitive inhibitor, lines shift counterclockwise, but it passes the same y-intercept. For non-competitive inhibition, here this line represents Michaelis-Menten kinetics without inhibitor. After adding a non-competitive inhibitor, this line shifts counterclockwise, but it has the same x-intercept. For uncompetitive inhibition, this line represents Michaelis-Menten kinetics without inhibitor. After adding an uncompetitive inhibitor, the line moves upward. Now, let's go to part 7. There are several methods to control enzyme activity, such as proteolytic cleavage, covalent modification, sequestration, allosteric regulation, and induction and repression. Among them, proteolytic cleavage, covalent modification, sequestration, and allosteric regulation acts quickly. Induction and repression results in change in amount of enzyme. It may take hours for the effects to show up. For example, trypsin is derived from trypsinogen, which is inactive. After removal of the first 6 amino acid residues by proteolytic cleavage, it becomes active. Now, this protein is called trypsin. Enzyme can be modified covalently, such as phosphorylation, adenylation, ADP-ribosylation, methylation. Today, we will focus on phosphorylation and ADP-ribosylation. As I mentioned to you in Chapter 1, phosphorylation occurs in serine, threonine, or tyrosine. And this reaction catalyzed by protein kinases. ADP-ribosylation is addition of ribose nucleotide on arginine of a protein. This reaction is catalyzed by a transferase. This has clinical correlation. For example, cholera toxin produced by bacteria Vibrio cholerae, ADP ribosylates the G protein GFS, leading to watery diarrhea. Pertussis toxin produced by bacteria Bordetella pertussis, ADP ribosylates the G protein GFI, leading to whooping cough. Allosteric regulation is the regulation of an enzyme by an effector that binds at the sites other than the enzyme active sites. So for simplicity, I only show you two subunits here. One subunit contains catalytic sites, one subunit contains regulatory sites. When the effector binds the regulatory sites, enzyme become either more active or less active. Allosteric regulation occurs in allosteric enzyme. Because they have more than two subunits in active sites, they do not follow the Michaelis-Menten kinetics. Effectors can be positive or negative. Positive effector is the one that increases enzyme activity. Negative effector inhibits enzyme activity. Homotropic effector is the substrate itself that serves as an effector. Heterotrophic effector are the one that is not a substrate. For example, IMP-GMP 5' nucleotidase use GMP or IMP as a substrate. Binding of one GMP molecule to a single subunit of the tetrameric enzyme leads to increased affinity for GMP by the subsequent subunits. So in this case, GMP is a homotropic effector. Another example, aspartic transcarbamylase use aspartic as a substrate. Binding of aspartic increases the enzyme activity. Therefore, it is a homotropic effector. ATP is a positive effector. CTP is a negative effector. Both ATP and CTP are heterotropic effectors. Allosteric enzymes do not follow Michaelis-Menten kinetics. They do not show normal hyperbola when graphed on a kinetic plot, but rather show sigmoid kinetics. Here is a kinetics for an allosteric enzyme without effector. And this shows sigmoid kinetics. This is because allosteric enzymes have multiple active sites. These multiple active sites exhibit a property of cooperativity, reminiscent of hemoglobin. Allosteric enzymes are influenced by substrate concentration. At high concentration of substrate, more enzymes are found in the R state or relaxed state. Conversely, at the low concentration of substrate, more enzymes are found in the T or tensed state. Allosteric enzymes are regulated by other molecules, which are called effectors. For example, positive effector stabilize the relaxed or R form. Activation is seen as the sigmoidal curve shifts to the left. Negative effector stabilize the tense or T form. Inactivation is seen as sigmoidal curve shifts to the right.